agrobacterium infiltration. Then we have then protein purification by extracting the total protein and using IgGs, separate beads, to purify the protein. Then protein macroarray, which involved hybridization and scanning using GenePix scanner. And finally, a statistical and bioinformatics analysis of data. This is the vector that we use, PYL436, which was used as a backbone. We cloned our DNA gene inside this vector using topocloning technique. And after infiltration of the Bentamiana leaves, and this protein was, this is the construct which was produced in the leaves, which had BAC4, MYC, histidine, 3 cleavage site, and 2 IgG. IgG beads were used to separate this product from the total protein, and cleavage enzyme was used to purify our protein, our construct. This is the final construct that was purified, BAC4 from whether tryptophan or Arabidopsis, then attached to a MYC and histidine. This was used to hybridize it against microarray chip, which are the proteins which had Arabidopsis protein library attached to it. Then we hybridized it with our proteins, which attached and interacted with the proteins. They interact normally and binded. Then we wash them to get rid of the unbinded proteins. Then we treated with anti-MYC IgG, which was our primary antibody. This identified the MYC and interacted. After washing, we had our secondary antibody, which attached to the, which was an anti-IgG conjugated with Cy5. This was, it had a fluorophore, which was Cy5, and this whole sandwich could be detected using special scanner. These are the actual results. We wanted to verify our protein has been purified properly and expressed. This is, we use STS page using composite blue staining as well as Western blotting. This is the total protein from Tryptophan multiformis and AT bag, Arabidopsis bag. Then we have the leftover from beads treatment, and this is our eluted solution. Our protein is there. We have this thick band, which is from the cleavage enzyme. It's unavailable, it's always there. Then we have our own protein, which is a faint band. Then we use Western blotting to verify it, and it was there, and we were very happy. It was very difficult to be a few months. So we use this protein, purified protein, to hybridize the slides or protein macroarray chips. These are the actual results or the actual scanned images of a scanner, which was a GenePix scanner. Each slide is covered with 5,000 proteins from Arabidopsis. Each protein has at least 2 replicates on it. Then there are positive and negative replicates, positive and negative controls on each slide. After scanning, we can see that the pattern is very similar, and it's expected because it's BAC4 from the protein, from the plant. Both of them are from plants, so they have similar patterns. Then the report file has been generated using GenPix software, and we used the two known proteins which we know already interacts with BAC4, HSC70 and UBA, and we use their signal to find the other proteins with the significant signal value. And we identified 407 proteins for Arabidopsis BAC4 and 671 for TIL-BAC, or Trichogonoliformis BAC4, which Trichogonoliformis had 266 interaction protein or protein partner more than Arabidopsis. When we looked at the pathways that these proteins were involved, we noticed that the majority of them were involved in carbohydrate pathways, such as sucrose degradation or glycolysis. Although we couldn't look at each one of them one by one because there are too many and it's impossible, but in general they are involved in carbohydrate. Now, for analyzing these results, we need to look at the autophagy cycle. When plant is under stress, this stress could lead to nutrients starvation or ROS production, which leads to sugar breakdown. This produces ATP, and other things ATP could be plant release this ATP actively to the outer space and makes it EATP. This EATP is a signaling molecule which can activate autophagy or apoptosis. In autophagy, which basically means self-eating, plant breaks down the proteins which has been oxidized or damaged, or the organelles that are not important. The proteins, such as the proteins, have been damaged by oxidation or misfolded proteins. Recycling the components and detoxification of the cell results in giving cell a chance to survive, basically. It's a recovery procedure. If this stress is prolonged, it can lead to PCD again. But basically, autophagy is a pro-survival pathway, while PCD is just detrimental, it kills us all. Most of the proteins that we identified are involved in autophagy pathway. So it seems to be reasonable to speculate that that BAC4 is involved or has its post-survival characteristics because it is interacting with proteins that are involved in this autophagy survival mechanism. Now, to summarize our molecular work, we can say that our molecular results show that BAC4 is probably involved in autophagy, and through that it is a pro-survival mechanism, pro-survival protein. Then BAC4 from Trichogonoliformis appears to have more protein interaction than compared to AT-BAC, and the influence of BAC4 on autophagy pathway is remained unknown because we still do not know how it impacts it. The results of high-density protein microarray provided valuable results for future studies, and the significance of this project could be summarized in a few points, such as this project for the first time provided characteristics of leaf structure of Trichogonoliformis in hydrated state, as well as structural changes during dehydration and rehydration. It also highlighted new roles for some of the structures, such as squamous tissue, as well as reform cells. It contributed to the knowledge of physiological changes during dehydration and rehydration, and it proposed that BAG4, for the first time this project proposed that BAG4 could be involved in autophagy, and through that it is a pro-survival molecule. And it also used Trypagon loliformis as a new resurrection model, and provided a valuable resource of high-density protein microarray that can be used with future work. And also, this is the first time that protein microarray has been used for a resurrection plant. There are some suggestions. Further structural studies need to be done on mitochondria during dehydration and rehydration, because it hasn't been observed properly in previous studies. And also the level of ROS during dehydration and rehydration needs to be measured, the actual ROS. And to validate the results of protein microarray results, further experiment needs to be done, such as yeast to hybrid, and further research needs to be done to clarify the exact role of BAG4 in autophagy. The results of some of the results of this experiment has been presented in 2 international conferences as poster, as Saba mentioned, International Gordon Research Conference in 2012, and in Interdrought 4 International Conference in 2013, and recently we submitted a paper to Physiology of Plant Hormone. I would like to thank first, I want to thank God, and then QUT. QUT for giving me the opportunity of studying here. Also, I want to thank Tribogon for, although it was really different. I want to thank QUT for giving me this opportunity to study in here, receiving tuition fee waiver. Also, I want to thank my supervisory team, Sabina Mundri, Tanya Shraskin, and Brett Williams. They were extremely helpful, and they were the best supervisor I could imagine. I want to thank my parents and Professor Akram Taji, that because of her I'm here now. I would like to thank my group, my stress group, Obiotic stress group, which was very supportive of me, and also our gang at 5. And thank you.
Thank you very much. I'll open the floor now for some questions.
Thank you. That was very interesting. I'm interested in the protein chips, because I haven't seen it very much done with those myself. Yeah.
It has been done only in 4 papers and plants.
And do the chips, are the proteins that are on the chip, are they just single open reading frame proteins, so they have proteins that include like multiple subunits, or are they still maintain their structure, and your proteins that you're using to probe the chip, do they also maintain their structure in that sort of format which you use?
What I read was that they only have the ROFs, open readings, and that's the important part that proteins interact. And from the results that we got, we knew that there are some proteins that interact with them. There are HSC70 and also UVA, and we noticed that they are present there. So it basically validates itself to say that the results were true.
Do you think there could be interactions that might be missed through?
It is possible. It is possible, definitely, because First of all, the proteins are dehydrated, so they change some of the structures that they change the secondary structure. It is possible that we lose that. That's why I mentioned that we need further verification. We need some more experiments such as yeast to hybrid, YIFC or other things to see really in the cell if it is working or not.
Question, please. Yes.
I think that was very important. So your particular plant, what makes it, based on your findings here, what makes it a best candidate to study desiccation processes? Because presumably you'd be targeting various characteristics for perhaps downstream application in an agricultural scenario. Have people looked at some of the characteristics previously in other desiccation type plants? And what is it that makes, from your work, makes it a better candidate than others?
The interesting thing about desiccation tolerant plants is that they can take the tolerance toward dehydration to extreme. So we want to understand how they can take it that far. And also there are other characteristics that are unique or present to this plant that are favorable, such as it is a part of actually polysis. It's a true grass, which is a member of grass family that most of the other stable foods such as rice, wheat, corn and other things exist. So probably it's easier to use these characteristics. For example, the structural characteristics, physiological characteristics that this plant has can be used to find plants, to select plants, lines from our crops that have these characteristics. Probably they can tolerate desiccation, dehydration, sorry, not desiccation, dehydration, or they could be more drought tolerant. Would you like to explain more?
I was just thinking maybe some examples. Have they failed in the past with other species?
Oh, they have.
Are they
No. Yeah, they actually didn't fail. There are many other, well, not many, there are a number of other resurrection plants have been used mostly from Southern Africa. They actually, for example, in one case, they found a protein and they transformed corn with it, and they were more drought tolerant. And it was in field trial, I'm not sure if it is released or not now, by Jill Farrens in group in South Africa.
Any other questions? If not, join me in thanking Parag for a really informative presentation.
Thank you.